Right. Okay. Good morning. My name is Meg Detloff, and I work for USGS PNAMP. Thanks for joining us for the PNAMP Fish Monitoring Work Group. Before we get started, I like to go over a few logistical things just to help you guys out if you're new to Teams or this in general. Unless you're speaking, please turn off your camera and your microphone by clicking the icons on your Teams platform. You guys can see that on the slide that's being displayed. If you have questions or feedback, please use the chat function, raise your hand, or unmute yourself. We can mute you, but we cannot unmute you. We're recording the meeting, and it will be available on our YouTube channel. If you're having trouble with the chat function, here are a couple of troubleshooting tips. Try leaving and joining again. Use a different browser. Edge works better than Edge works better for Teams rather than Chrome. Or just try using the desktop version instead of the browser or vice versa. If you don't have access to the chat, please feel free to unmute yourself and comment or ask questions. That's not a big deal. Let's see. PNAMP commits to fostering an open and welcoming environment, free from harassment and discrimination, so please remain respectful. I would like to acknowledge the Fish Monitoring Work Group leadership. Names are associated with the tasks they have previously or currently lead. Our leadership is partially based on in-kind time, meaning these folks saw a need and were willing to use their time to help solve a problem, and we are very grateful for their help. The core team consists of Kasey Bliesner, Marika Dobos, Laura Erickson, Nancy Leonard, Russell Scranton, Mari Williams, and Jen Bayer. To help me keep track of participation, please open the chat and introduce yourself with name and organization. This also helps you acquaint yourself with the chat function. While you guys are doing that, I will quickly cover today's agenda. We're going to kick the meeting off with updates on current tasks. Next, Kasey, Ian, and Melody will present key insights from the Middle Fork John Day River IMW 2024 summary report, followed by a little bit of Q&A, and then we'll close the meeting with a little wrap up. But before we jump into task updates, I just wanted to chat with you guys about the 2024 Fish Monitoring Work Group Survey. Thanks to everyone who provided input. We are listening. Overall, we had very positive feedback on the helpfulness of this group. No negative input at all. Highest preference for presentations go to research results and lessons learned. So this kind of checks that box a little bit with IMW summary. Then emerging tech, followed by analysis tools, and then data management. There were great suggestions for presentation topics, including artificial intelligence, new tech for watershed scale monitoring, kind of like IMWs, bull trout pop monitoring, application of fish monitoring data and policy or related programs, such as like water transaction effectiveness, integrating remote sensed and field-based observations, and the tricky dance of data management and data governance and how this folds into research results. We'll also consider presenting task updates in a different format. We're doing this a little bit today, possibly doing maybe a more in-depth highlight on one specific task during a meeting. So we'll kind of mull that over for 2025. Again, thank you for your feedback. And if you didn't get a chance to do the survey, Jen's going to drop the link in the chat right now. So if you guys want to do it, it's still open. And if you're not sure if you did it, just let me know and I can kind of look if you provided your name in the survey.
I just dropped it in the chat. There you go.
Thanks, Jen. And Jen, like, let me know about comments because I can't see anything right now. So for task updates, these are overviews of progress. If you're interested in learning more about a particular task, visit the project page on the PNAMP website. Jen's going to drop that link too, and just select one of the tasks. Leadership of the task will give an update if they're here today, and I'll briefly pause for questions and comments. If you have a question after we move past a task, just type it in the chat and Jen can help me circle back after all the updates are complete. And then Russ, I don't know if you're out there.
I am.
Feel free to turn your camera on. Yeah, so Russ is going to cover the carrying capacity update for us today.
Can you hear me or no?
Yeah, I can totally hear you.
Okay. Okay, just as a quick overview, there is a process where the team, Morgan Bond, Tim Copeland, Tim Beachy, myself, Jen Bayer, have been working with additional co-authors to produce a paper on carrying capacity standards. The additional co-authors are Joe Benjamin, Ryan Belmore, Kevin C., Corey Green, and Meg Deploff. You've also been helping us. The basic principle is to provide a paper that shows different products and different models and expansions that produce carrying capacity outputs, how they vary and what the significances of those are, and what some best practices may be and how to manage that information. The specific expansions we're looking at include habitat expansions, stock recruitment models, quantile regressions, individual-based models, and food web modeling. Currently, we have a draft document. The authors are editing each section to match tone and brevity. We're editing a final discussion, sections, tables, and graphics and figures. This is an example figure that's been updated over time of how the different products may produce outputs. Our next steps, as I said, are continue to adapt these figures based on author feedback and get the full document out for external review and seek eventual publication. Next slide, if there's one.
No, it's just this one, Russ.
Yeah, that's what I thought. Any questions?
Jen, let me know if anybody types anything in the chat.
I don't see anything in the chat.
Okay, groovy. And like I told you guys, like if you come up with something or we're too speedy, just type it in the chat and we'll circle back before we move on to the presentation. All right. So next up, Megan Griffiths is of PSMFC, is currently leading this part of the task. She provided some updates for me to share. She was unavailable to be here today. StreamNet has a dashboard in development that will contain specific information from multiple regional systems and sources for rotary screw traps in the basin. Active development will begin in the coming months, funding dependent, with the goal of presenting it to the Fishmeyer work group for QA/QC of the data in December. The dashboard is estimated to be fully functional by fall of next year. Other items of discussion that are being kicked around are non-basin data and whether it can be integrated into the dashboard. Do you guys have any questions? And I'll try to field them the best I can. And if not, the best course of action is just reach out to Megan. And her email's listed on here too. Or you can just reach out to me and I can connect you. Pit tag array data and related data analysis is focused on documenting and recommending improvements to data management and analytical methods and tools for pit tag array data. At the end of January, Marika Dobos presented the task, and we pulled participants at the Pitagus workshop. Participants provided valuable insights and feedback, which we homed in on with the 2024 Fishmeyer work group survey that I talked to you guys about earlier. Standouts from the survey were a need for a one-stop shop to help point people to resources for pit tag data and analysis, a series of pit tag specific webinars targeting analysis tools, data cleaning and prep, and how-tos for downloading from data repositories. The workshop, which came in third, would target more in-depth trainings on analysis methods. So we have begun planning a webinar series to address some of these requests. There are two components to this: tools to improve effectiveness for managing pit tag data. Wowzers! And then number two, analyzing detection data from pit tag arrays. We first want to provide good tools at all skill levels to streamline data management and formatting for specific needs. We also want to introduce data tool repositories where these tools can be found. After the series, we'll see how much interest we get and then can transition our objectives towards data analysis, which would likely be the hands-on workshop. We kind of feel like with the analysis part, you need to be in the room with your data or a certain data set and working through it with a group of people on your laptop. This slide is the potential structure of the webinar series. We are currently in discussions with presenters and reaching out to other experts. We've discussed it and shifted it again and shifted it again. So I saw you were out there, Ben. So we're still chipping away at this, but super stoked to have you on for GitHub and then some other folks. So if you guys have a request, please feel free to contact me. We can add something in here if you're like, this is a need I'm seeing in my agency or organization, and it would be really helpful to have somebody present on this particular thing. Does anybody have any questions on this? And we're kind of hoping this will kick off in February or March of 2025. Jen, you're my eyeballs.
Russ, go ahead.
In addition, I think one of the things on the training and tools for repositories, besides just talks, one of the things that we're talking about is trying to organize or improve kind of a list or repository of tools. An example is that UW CBRs has a bunch of tools. Dart has a bunch of tools. There's Pit Cleaner. There's the Pit Hyper tool from One Fish, Two Fish, ways to clean data, but they're all disparate. And so is there a better way to make people's known tools and code available for managing pit tag data? So I think that's one of the topics we'll also present, but I'll try to host kind of a little bit of a clearinghouse of these analytical tools that people could be using, or trying to make recommendations of how to manage that. So your feedback on that would be very helpful, because we know there's lots of R code out there and different ways that people have programmed their own analytical tools. But we just want to try to stop reinventing the wheel and share the expertise in the basin.
Totally. Well said.
And I want to say that we know that in the Columbia Basin, Pitagus has a bunch of stuff, a bunch of resources on their website too. There's some webinars, there's some recordings, there's various things. So we're not reinventing the wheel, but some people haven't found those. So the idea of a one-stop shop will help lead you to a variety of resources. And also, we also want to acknowledge that there are folks outside the Columbia Basin who are using pit tags, of course, and there are systems emerging for, broad scale sharing of data and whatnot in Klamath and in the Salish Sea. And so, we're trying to provide resources training that might help everyone. And we're trying to sift through that. So Meg mentioned Ben, we mentioned Marika Dobos, who couldn't join us today. There's people who've raised their hand already to help shape this. If you really feel like you want to help shape it, speak up soon, because like I said, we're trying to put this together so we can fit in the timeline that you all said works best for you, which is February, March, which is not very far off. So if you really think, gosh, I want to help shape that, let Meg know, or drop us a note in the chat now, because we're trying to move this along so we can implement it this winter.
Totally. Perfect. Well said, Jen and Russ. Thank you. Questions on the task? All righty. All right. Jen and Mari are now going to talk about the most recent task added to the Fish Monitoring Workgroup lineup, updates, terms, and definitions used in CAP data standards. Hand that over to you, Jen.
All right. Are you out there, Mari? I know Mari was.
I am.
Might be late. You want to start?
It looks dark. No, you go ahead. Jump in.
Okay. All right. So for CAP aficionados, you recall last year we had a big workshop. One of the things that came out of the workshop was, maybe there's a little confusion on terms and definitions and how do we access that level of detail that is part of the data standards that StreamNet, for the data systems that StreamNet hosts. So there are definitions and they are documented, but there was definitely some questions, and there's a lot of new biologists out there from the time that we built those systems quite a while ago, not aging or any of us. So we want to update that. We heard this from you all that it's important. So next slide. as part of the update, what we're doing is reviewing. So I think I kind of talked about this. One of the things that's new, though, is we are having conversations with a pretty broad scale lately, an international scale, about connecting control vocabularies for salmon data internationally. So our work here in the Columbia Basin is frankly ahead of everyone else, I'll brag, because we have this cool community of practice where people have weighed in and built systems together. And so we want to share that with others and think about how we can all benefit. And again, we want to make sure our stuff is first sound and we all agree on it, and two, that it's accessible. So this exercise will help with both of those things. Next slide. So our process is we have extracted the existing terms and definitions. Thank you, Mike Bannock. And Mari and I have waded through that first, and then we've had some other folks very active in the StreamNet steering committee who are very familiar with this work. We're asked them to go through and also review and flag. Once we're done with that, we've sifted through it, and we have some things that are obvious fixes. We want to align it with an existing system like Patagas, for example, or the ARMIS to make sure they're consistent. There was some minor wording thing, like things that we don't really need subject matter experts to review. That's the easy stuff. We are seeing some things that we want biologists to weigh in on and make sure that we all agree that this term has this definition, and we're all consistent about that. So that's the next phase of this. I think we have one more slide. And Mari, do you want to jump in on the process of how we got here? Did I forget anything before we get to what we're doing next?
I don't think so, but I want to make a mention that we, in coordinated assessments, built an imperfect system. We built a system. We knew it was going to need adaptation, and we built it that way. So this is a continuation of our original purpose within coordinated assessments to continue trying to improve upon it, not a outright critique of how things were done 15 years ago, because we built a great foundation to continue to grow on. So after Jen and I went through what Mike put together, we did send it out to other folks to try and see if they saw different flags within the terms and definitions as they currently are, and trying to condense down what we are actually going to review. So on the 4th, November 4th, we have a meeting with those that have volunteered, and anyone else that wants to join is welcome to come and give us your gut instincts on which areas we should be sending to subject matter experts, or how we interpret that. We are going to try and get a final draft done by January so that we can use it in the March workshop for the WABC AFS up in Vancouver next year. And looks like Dan is on board with joining us.
Thanks, Dan. We see
awesome. That. Jen, fill in any
holes.
We'll
share. No, I think that's the key thing is some of you've been hearing about it. We've been talking, we're going to do this. We are doing it. We've been doing it behind the scenes. November 4th, we'll have a kickoff meeting. We would love to have subject matter experts join us. We have not sent a calendar invite or information about that. You'll see it pretty soon. We're going to invite the FISH work group and the CAP community of practice, which there's a lot of your names are on both of those lists. We're going to send a broad invitation. We're hoping some of you show up to help us. If no one shows up, we're going to find you another way, because we really need to be able to say that subject matter experts are the ones who reviewed these terms. So we'll figure that out. We're striving for January. The March workshop Mari mentioned is something you'll hear more about soon. We're proposing a workshop at the Washington, British Columbia AFS chapter meeting to focus on how can we share data more broadly. Back to that international conversation I mentioned earlier. There are a group of folks working on how do we stitch together control vocabulary such that we can have an ontology that will bridge an international universe of people who are trying to help us advance salmon recovery and other things as well. So more on that later. We're about to submit our abstract tomorrow. So once it's accepted, we'll gladly invite you all to that as well. All right, I think that's it, unless there's questions. All right.
And obviously questions on all the tasks at this point, because we're going to lead into the presentation after this.
Yeah,
you guys are last. Save the best for last, Jen. Come on.
Oh, yeah, right. I think they're here to hear about this cool presentation from FMWG.
Definitely.
Thanks.
All right. Without further ado, I am honored to introduce Kasey Bliesner, who works for ODFW and is the co-coordinator of the Middle Fork IMW. The floor is yours, Kasey.
Excellent. Thanks, Meg. Hi, I'm Kasey Bliesner. I am a project analyst for the Oregon Department of Fish and Wildlife out of our East Region Fish Research Office. I also am the co-coordinator along with Ken Fetcho with OWEB for the Middle Fork John Day River Intensively Monitored Watershed. Today, I'm going to be presenting with Melody Feden, who is our assistant project leader on the Middle Fork IMW and also the acting John Day Fish Research supervisor, and Ian Tattam, who is the acting program manager for East Region Fish Research. We have a kind of a dense presentation, so I'm going to ask just kind of for the flow, since there's 3 of us presenting, if you can save your questions for the end, but knowing that some, I'm forgetful too. So if you have a question that pops up, go ahead, you can put it in the chat. Just for our sake, try and reference the slide if you put the question in the chat, and we'll come back to it at the end. All right. Meg, next slide. So the Middle Fork John Day River is located within the John Day Basin and is a tributary to the North Fork John Day River. And a little background, the John Day River is the 3rd longest free-flowing river, so undammed in the lower 48, and there's no hatcheries in this entire basin. The Middle Fork originates in the Blue Mountains, and the IMW area is located in the upper Middle Fork John Day Basin, so that section there outlined in brown. And the history of the Middle Fork is like that of, many of the Eastern Oregon, the Eastern watersheds. And the history includes ranching, cattle grazing, and mining. And you can see that picture from the Oregon Mining Review of the dredge. And so, portions of this basin were dredge mined. And that dredge is still located in the town of Sumter, if you're ever out in this area and want to go see it. And current land ownership is largely public. So all that section in green, so that's largely Forest Service owned. And there are a couple of conservation areas that are owned by the Confederated Tribes of the Warm Springs, and a recently acquired property up in the upper headwaters that is owned now by the Blue Mountain Land Trust. The anadromous species in this basin include lamprey, bull trout, and the focal species for the IMW, spring chinook, and threatened summer steelhead. Next slide. So the Middle Fork John Day River Intensively Monitored Watershed is part of a larger network of long-term, large-scale effectiveness monitoring projects called Intensively Monitored Watersheds, or IMWs, that are located across the Pacific Northwest. And intensively monitored watersheds are long-term projects designed to deepen our understanding of how fish populations respond to habitat restoration. The Middle Fork was established in 2008 by the Upper Middle Fork John Day Working Group. The current partner members are depicted in this fish infographic there in the middle, and consist of state and federal agencies, some universities, some NGOs, and tribal entities. we meet monthly with a core group of about 10-ish people, 10 to 15 people that really participate regularly, and a much broader, larger group that participate and contribute to the IMW. The base funding for the IMW comes from NOAA and is coordinated, NOAA and PSMFC, and is coordinated through OWEB. And so the specific goals of this IMW are to evaluate the overall benefits of restoration actions to summer steelhead and spring Chinook salmon in the upper Middle Fork John Day River, and to help understand how specific restoration actions impact in-stream habitat, water temperature, and cell-modded metrics at a watershed, sub-watershed, and reach scale. Next slide. So within this Middle Fork IMW framework, and to meet the goals of recovering the watershed, we need to understand and address limiting factors. So ecosystem diagnostic and treatment models identified habitat diversity, sediment load, temperature, and summer rearing habitat quantity as limiting factors for summer steelhead, and habitat diversity, sediment load, habitat quantity, temperature, and discharge as significant factors limiting Chinook productivity in the IMW area. So the three pictures show different locations located within the Middle Fork area that demonstrate, habitat conditions or things that have limited or contributing to those limiting factors. So you can see the Oxville Conservation Area there prior to restoration, showing that history of dredge mining, that channelized system. There were many log weirs spanning, parts of tributaries in the Middle Fork, including Camp Creek, and then, incised channels and trampled vegetation there. And so, further research and monitoring, really continues to show us that water temperature is the primary limiting factor in the Middle Fork, and Melody will have more to say about that later. And so the infographic on the right shows this interplay with limiting factors and demonstrates a hierarchy. And so this isn't necessarily a quantitative ranking, but a concept to help guide actions into addressing the most important limiting factor first, and then addressing others once the primary limiting factor is controlled. And that's something we've learned and others have learned within the IMW process. And so this down arrow indicates a hierarchical nature of the limiting factor. So changing habitat diversity will have limited effectiveness if stream temperature remains too high. And the curved arrows indicate interplay between factors. And so we recognize that the system is much different state than historic, and massive, structural changes or treatments to restore ecological processes may be needed in some reaches to effectively deal with water temperature. Next slide. So what has happened restoration wise in the Middle Fork IMW area? And so restoration has been ongoing over the last, 20-ish years. And a majority of the work has been focused in some very specific areas due to, landowner interest. And these orange arrows depict those important locations. Like I said, there's these three conservation areas owned by the Confederated Tribes of the Warm Springs, the Dunstan, Oxbow, and Forest. And these conservation areas represent thousands of acres and miles of main stem that provide opportunity to design and implement restoration actions. And these former cattle ranches were acquired with federal and state funding and have been key to providing restoration opportunities. And the Confederated Tribes of the Warm Springs are a very important and very active partner within the IMW. Another important area is the Camp Creek watershed, where the Forest Service developed a long-term restoration plan with input from partners, and has actively worked over the last 20 years to implement restoration actions along this very important tributary. And lastly, I already mentioned, but an important headwater property was recently acquired by the Blue Mountain Land Trust near the town of Bates. And this will also provide opportunity for restoration on a formal cattle ranch in the headwaters of restoration strategies have shifted a bit over the last 5 years. And so it's really hard to see in this map, but each of those different colors and shapes represent the primary restoration action that was taken in that area or for that project. And so as you can see, there was 149 restoration projects that have been implemented from 2008 to 2023. Most of these have addressed multiple limiting factors. There's been over 73 miles of in-stream habitat treated, over 250 barriers removed or improved, providing access to, I don't think that was supposed to be 135 stream miles. I think that's incorrect. And then in-stream water leases provided over 6 CFS of late season stream flow and riparian planting and fencing along 39 stream miles. And so, the shift that we've seen is from 2008 to 2016, restoration actions primarily included channel reconfiguration, so like fixing some of those places where the dredge mines and railroad grades and things like that have really straightened the channel, and to provide those fish passage improvements, so removing and improving those 250 barriers. In the more recent years, 2008 to 2023, restoration projects have focused on in-stream habitat improvements, really including a lot of thought about tributary reconnection and floodplain reconnection, and focused and prioritized riparian planting to start, addressing water temperature. But evidence from IMWs across the Pacific Northwest have demonstrated that very large-scale and extensive restoration needs to occur to move the needle on recovering our focal species. So while restoration has been widespread within the Middle Fork, we determined in this 2017 to 2023 timeframe, where we had really good GIS data tracking of restoration projects, that only about actually 17% of stream miles, so that's a steelhead distribution stream miles, have actually been treated. This is a big watershed, and more extensive and intensive work will need to occur for us to see a detectable response. Next slide. So part of the framework and part of what's inherent in IMWs is documenting and sharing the lessons. So, that's part of the reason we're here. I've called this our book tour. This is our fifth presentation. So trying to share what we've learned out of this Middle Fork IMW. So, documenting and sharing these lessons learned from the network of IMWs will help implement further restoration with greater confidence and provide effectiveness monitoring that can be prioritized and directed for maximum value. So this came as a quote from Bennett et al. In 2016. So how are we doing this in the Middle Fork IMW? So we initiate, the Middle Fork was initiated in 2008 with this 10-year time frame in mind. So in 2017, we completed a 10-year summary report. And as part of that 10-year summary report, we recognized that we would need to, put on our calendars, we need to do this again in 5 years. So here we are in 2024, completing this 2024 summary report. This kind of group requires, and doing this requires a lot of dedicated coordination and communication to both put these reports together and then share them out. And so we had a lot of organization, a lot of meetings, a lot of writing. It took us almost 2 years to kind of get this all wrapped up, or a year and a half to 2 years to get this all wrapped up and send that. And we had 8 partners who volunteered to write up their research for inclusion as chapters in the summary report. And it really built off of what was learned in what was written and learned in the 10-year summary report. And those 8 chapters included information that was either from ongoing projects, ongoing data collection, like water temperature loggers, watershed scale fish in, fish out, macroinvertebrates, or projects that were new as of 2018. And so Melody will talk about some of those fish projects. So there was 4 chapters that were really related to fish monitoring research. The other 4 chapters that were included were on macroinvertebrate community analysis, long-term habitat and vegetation monitoring in response to active and passive restoration, and a chapter on planting, efficacy, and groundwater monitoring. And so that QR code up there, if you scan that QR code, that will take you to a data clearinghouse record that will give you access to the 2024 summary report and previous IMW reports if you want to read through all of them. In addition to the research, and as part of an adaptive management framework, we performed an assessment of learning and recommendations taken. And we also asked partners for any new lessons learned and recommendations over the last 5 years. And so I'm going to run through a few quick highlights of this adaptive management assessment and some of the themes around planning and monitoring lessons learned and recommendations. Next slide, Meg. So an important piece of the Middle Fork, includes elements of adaptive management. So in 2017, when we wrote that 10-year summary report, we asked restoration practitioners and researchers for lessons learned and recommendations in 3 categories, including planning, monitoring, and restoration. And so that we could ensure that learning and best practices were documented and presented for incorporation into future actions. We received 86 recommendations, paired lessons learned and recommendations from that 10-year summary report. So for the 2024 summary report, we asked a core representative group of IMW partners to evaluate those 86 recommendations and, evaluate for us, were they addressed, partially addressed, not addressed at all, or no longer applicable? And overall, partners felt that about 86% of the recommendations made were either addressed or in this partially addressed / ongoing. And so that really indicates that, we are communicating and communication is happening among partners, and it's been mostly successful, and that recommendations are recognized and efforts have been made to address them. And so that full list of 10-year report recommendations and categorizations can be found in Appendix B of the 2024 summary report, if you want to read through all those. And so as part of the 2024 summary report, we did this assessment of previous recommendations, and then we asked for new recommendations, and we received about 67 new recommendations. And so for the next couple slides, I'm going to share some themes around these new recommendations and then give you some examples of how, how recommendations from the 10-year summary report were incorporated into, monitoring and planning. All right, next slide, Meg. So for this example on adaptations in planning, the challenge, or what we heard was that communication between restoration and monitoring practitioners was not consistent across all efforts. And so the lesson learned was, that the IMW shows that continued coordination is needed, especially when multiple agencies are implementing a variety of monitoring and restoration actions across a broad area. So you saw that efficient infographic with all those different agencies. And it's also challenging to compile all the information needed for the varying, restoration inventories and data inventories and summary reports. And so starting in 2018, we started hosting annual joint meetings with the John Day Basin Partnership, which is made up of primarily restoration practitioners. to improve communication across monitoring and restoration efforts. And then additional restoration coordination meetings began being held quarterly as needed. And so these really helped collaborators, learn where monitoring and where restoration were occurring and where actions need to be planned for the future. All right, next slide. So from the 2024 summary report, when we asked for lessons learned and recommendations, new ones, these were some of the themes that we, came up with. So, they're not prioritized, but we did kind of go through and read all of them and look for these themes. So, again, more communication between research, monitoring, and restoration planners, clear and direct recommendations for restoration planning, permitting, and communicating with partners, pursuing time and money for further analysis and publication of results, develop and maintain an archive of IMW-related publications, investments in long-term stable funding for monitoring and outreach and demonstration to out-of-base practitioners, what, who, and how to. So that's, we're already implementing some of these recommendations by giving this presentation today. All right, next slide, Meg. So for adaptations in monitoring, one of the challenges that came across was, that despite some increased spawning habitat capacity, we haven't observed a discernible watershed scale response to restoration efforts within the first 10 years of monitoring in the IMW. So, the lesson learned, change at the watershed scale takes a long time to realize, and monitoring, needs to occur at a matching reach scale where changes from restoration can be detected. And so the adaptation to that were that monitoring efforts were increased to quantify the spatial variation in Chinook salmon and steelhead productivity metrics and habitat variables across main stem rearing habitats. And in addition, those were linked with productivity metrics and water temperature to identify areas that were disproportionately impacting populations. And so Melody will actually give a much more detailed results from that adaptation and that monitoring effort later in the presentation. All right, next slide. So for monitoring, some themes we heard for recommendations for future monitoring are to produce studies that provide actionable results for restoration practitioners. So, not just saying that, giving fish results, but saying like, here's what needs to be done to, improve productivity or to provide this habitat metric. Expand monitoring in the tributaries, expand monitoring that includes hydrologic monitoring and more specific and intensive temperature monitoring, looking for thermorefugia and central temperature zones, et cetera. Physical habitat monitoring and assessments, including a watershed assessment, and tools to utilize drones and drone imagery and some of this remote sensing to assess restoration effectiveness. So that that wraps up my piece of the presentation, giving the background and some steps that we've been following with adaptive management. And so we're going to shift gears into monitoring, and Melody is going to dive into some of the key insights from fish monitoring and research in the Middle Fork. Melody?
Awesome. Yep. Thank you,
Okay.
Kasey.
Excellent.
Meg, will you advance to the next slide, please? And I just want to make sure like everyone can hear and see me. I've been having some technical difficulties this morning, so I hope that it remains good for the next 15 or 20 minutes. So Again, thank you very much, Kasey, for that background and information. And I just wanted to, first of all, thank everyone for coming to this meeting today and inviting us to speak. We're really excited to share these results with you today. So here on this slide is an inventory of our monitoring projects that have occurred in the Middle Fork since we started keeping track of these records. As you can see, these monitoring projects have covered a wide variety of topics, themes, dates, and locations within the Middle Fork IMW. The breadth of monitoring that has occurred in the Middle Fork is truly impressive. We've counted 38 separate monitoring activities that have occurred since 2006, and this is most likely not even a comprehensive list. So it would have been impossible to report on all of these projects on our most recent summary report. Luckily, several of them were reported on during the 10-year report. So the projects that are highlighted in blue on this list are projects that were included in the summary report, and then the ones in the yellow indicate specifically the fish projects that I'm going to go into more detail today, which we thought were the most appropriate for this audience and this group. Meg, next slide. So the first project I'm going to talk about today is our, the first project I'm going to speak about today is our watershed scale Chinook and steelhead abundance and productivity monitoring. And for these projects, we estimate our spawner abundance of steelhead and Chinook salmon adults in the Middle Fork John Day. We also look at our freshwater productivity of Chinook salmon and steelhead populations. So these monitoring projects are what I consider the backbone of our fish monitoring in the John Day Basin. It's where we really can study and report on our fish in and fish out on a watershed scale. Next slide, Meg. So the methods we employ to look at these metrics, first for spawner abundance, we use red surveys on spawning grounds. And for steelhead, that includes grit surveys because the spawning ground, the range of spawning in the basin is so extensive. For Chinook, we're able to complete census surveys where we actually walk all of the spawning grounds to measure the abundance of adult Chinook. For freshwater productivity, we use rotary screw traps, which are employed on the Middle Fork, Main Stem, and South Fork. And for these basins, our main stem is used as a control for our Chinook populations. The South Fork is used as a control for our steelhead, and those basins are also controls for our adult populations. And next slide. And I wanted to just set up kind of what we were expecting to see from the beginning of when we implemented the IMW. What we expected to see between our treatment and control reaches. Control reaches are ones that have a lot less restoration implemented than the Middle Fork, which has extensive restoration. What we expected to see in these populations was for them to kind of track over time. And what we were hoping to see was that Middle Fork population start to outpace the control population due to the amount of extensive restoration in the Middle Fork. Next slide. So that brings me to some of our actual results of our Chinook spawner abundance. And you can see that we have time here on the x-axis with the spawner abundance on the y-axis. And interestingly, this does look a lot like our expected results where our treatment and control basins track with each other over time. But as you can see, what we're not seeing is we're not seeing that response to restoration that we are hoping in these adult populations. Next slide. So some more results from our freshwater productivity, and this is looking at steelhead. Again, a similar graph where we have time on the x-axis and productivity of our out-migrating smolts on the y-axis, and we have the South Fork as our reference and the Middle Fork as our treatment population. And this is where as you can see, like those expected results, these two basins are tracking through time. But you can see that in 2015, the South Fork Basin, the productivity takes a dramatic decline in the downward direction. But for the Middle Fork, it doesn't decline as rapidly, and we're starting to see some stability. We're not calling this an ironclad response into restoration, but we are keeping an eye on these trends. Next slide. So our takeaways from these watershed scale monitoring are that so far we have not observed a discernible watershed scale response to restoration, but while we haven't seen productivity increases, we have seen stability. For our future monitoring with regard to these projects, we will continue to monitor our fish in, fish out. Our steelhead surveys are most likely going to be moving to a pit expansion method versus spawning ground surveys. And since we're not seeing this broad scale population recovery, kind of like what Kasey set up for me really well, we have developed other monitoring projects looking at smaller scale responses to restoration with the hopes of using adaptive management to discover and promote restoration practices that are most effective. And while compiling this presentation, some themes emerged from while I was reviewing the chapters. So my next sections are not necessarily covering specific chapters of our report, but kind of some overarching themes that we discovered along the way. Next slide. The next project that I'm going to talk about is patterns of spring Chinook salmon dispersal. And some of the objectives of this project include we were trying to describe dispersal patterns of Chinook during the fry and par life stages. We wanted to identify environmental factors that are driving these dispersal patterns. We also wanted to gather information on possible effects of shifting red locations, which is something we did see in response to restoration in the Middle Fork Basin. Next slide. Okay. So our methods for this project first started where we collected genetic samples from adult Chinook carcasses. We then later on in the life cycle, we collected genetic samples from the fry and par offspring from those adults. Using those genetic samples, we were then able to match offspring to female parent that allowed us to determine an approximate origin location to calculate dispersal. Next slide. So here are some results from our fry dispersal project. We have dispersal distance on the x-axis in kilometers, and then we have fry density on the y-axis. And we found with the fry life stage, there was no upstream dispersal detected. Most of the fry that we captured did not disperse greater than a kilometer from where they originated, but many still dispersed a great distance. You can see from this graph that approximately 25% dispersed up to 5 kilometers, with some dispersing up to 20 kilometers. We're still assessing some of the drivers of fry dispersal, but we hypothesize that is related to flow conditions and habitat availability. The habitat where we most commonly found fry were inundated floodplain habitat that are adjacent to the main stem that were usually included vegetative cover. Next slide. We also were able to look at dispersal at the par life stage, and these graphs are set up in a similar fashion to the fry dispersal with the dispersal on the x-axis and abundance on the y-axis. I'm going to first direct you to the graph where we had all the par grouped together. And we found that there were some similarities with the fry life stage, but we also found some differences. We found that still most par dispersed in the downstream direction from where they originated, but approximately a third of par were able to disperse in the upstream direction. So we further broke those up into two different categories, into par that dispersed in the main stem and par that dispersed into tributaries. And here we noticed that for par that dispersed into tributaries, that's what encouraged some of that upstream dispersal. And we think this is most likely related to temperature, as in the summer, the tributaries are much cooler than the main stem by several degrees. Next slide. So this is another way to look at our par dispersal results, and it shows where fish were born and where they dispersed to. And I know this graph has a lot going on, but I'm going to hopefully slowly walk you through it. So we first have fish that stayed in the main stem, that's represented by red on the right, versus fish that dispersed into tributaries, which is, sorry, red is on the left, blue is on the right, into tributaries, blue. The black line on all of the graphs is the mean daily maximum July temperature of the main stem, which you can see is actually quite variable. So the gray bars show where the fish originated from, and the red and blue colors show where they went and how abundant they are at that location. And what we found interesting about this graph was that for most areas of the river, fish in that area originated from nearby. One of the unique situations is in the Oxbow, where some of the most intense restoration has occurred in the Middle Fork IMW. And that is, you can see on the graph, it's around kilometer 91 to 97. So you can, if you look in the red in the main stem, you can see that several fish came from the upstream direction and the downstream direction, and sometimes dispersed many, many kilometers to be in that location. The second takeaway that we noticed from this graph is that most fish generally disperse from areas of warmer water to areas of cooler water, and that was either in the main stem or into a tributary. Pretty much any dispersal into a tributary was going from a warm to a cooler location, but that also was exhibited in the fish that dispersed to other locations in the main stem Middle Fork. Next slide. So some of the major takeaways from these projects are that we're seeing widespread dispersal at both life stages, more than that has been cited in literature, and we thought that that discovery was exciting. Restoration aimed at the fry life stage would be most effective 1 kilometer downstream of dense red locations, so that kind of restoration would include a well-connected floodplain with riparian cover. Our other takeaway is that tributaries encourage upstream dispersal in ways that we're not seeing other like upstream dispersal occur. We also learned that shifting red locations could change access to cool water areas and tributaries. If the red location changes into a hotter area of the river, if most fish are only dispersing up to 1 kilometer, that could change their access to these cold water refugias. Next slide. And the last topic I'm going to talk about today was a couple of different projects where the objectives were to describe juvenile abundance and distribution across the rearing habitat and to investigate drivers. Also to quantify spatial variation in juvenile cell-mounted productivity and survival. Additionally, evaluate the relationship between juvenile cell-mounted productivity metrics and habitat variables. Next slide. And I'll say this project was also done on the Chinook juveniles, not the steelhead. So our methods for these studies were we conducted snorkel surveys across rearing habitat and modeled abundance using detection efficiency and habitat metrics by a model developed by Staten et al. 2022. We also used pit tags, conducted mark recapture studies to estimate abundance, growth, and survival from restored and unrestored sites. We also collected several habitat metrics using a pared down version of the CHAMP protocol. We also have temperature loggers deployed across the basin. Next slide. So here are some of the distribution results. We have river kilometer there on the x-axis with our main stem par density on the y-axis, overlaid with our July mean maximum daily temperature. And what you can see from this graph is there is an inverse relationship between water temperature and par abundance. In areas with high temperature, we see very low par abundance. And so we use this data to build a linear model that showed there was a significant relationship between temperature and par density. When maximum July temperatures start to reach 23 to 24 degrees Celsius, we see a very low density of approximately 0 to 1 fish per meter, compared to some cooler areas that contain up to 5 fish per meter. Next slide. Some of our other survivorship results. Here we have river kilometer on the x-axis with survivorship on the y-axis. And this graph, we have the blue shaded areas are sites that are used as controls or non-treatment sites, with the brown shaded areas are our treatment sites. And what we learned from this graph is that we couldn't find a significant difference between treatment and control sites, but there was a strong relationship between survival and temperature. Meg, will you click one more time? There'll be one more graph that populates. Thank you. So we did see that there was a strong relationship between survival and temperature, with higher survival apparent at cooler maximum temperatures. So for us, this verified that temperature is a limiting factor in the basin, and we can't expect to see population level responses to restoration until high water temperature conditions are improved. Next slide. So to summarize that, again, some of our takeaways are that temperature is an important environmental condition driving distribution. Survival to the smolt life stage has a negative relationship with increasing water temperature. And in areas where temperature is relatively cooler, we run into these density-dependent factors like growth that also become limiting. So in those areas with that have cooler water, all the fish are attracted to that area and then are limited by how much they're able to grow in those areas. Next slide. So through these projects, we developed a map that has several restoration recommendations, but this is where I'm going to pass the baton off to Ian, who's going to walk us through this figure and to help wrap up the presentation.
Awesome. Thanks a lot, Melody and Kasey. Thanks for the tremendous background and wrap up here. Hopefully everyone out there in cyberspace can hear me. Someone chat or something if they can't. I'm going to try to distill down some of the recommendations here and connect them to actions on the landscape, and then provide a little bit of overall context for where we're at in the Middle Fork IMW, what we've learned, and where the populations are at in sort of their recovery trajectory, or the hopeful recovery trajectory. And then we're going to open it up for questions and discussion. So first of all, in this figure here, orient everybody to where we are. This is, again, the Middle Fork John Day, and it's flowing from the bottom right of your screen to the sort of the upper left of your screen. And what we've done here, I should say we in the colloquial sense of Lindsay Sapello is really the lead on putting this together. We've tried to color code the different reaches into the primary current limiting factor to help guide restoration action. So the red reaches are areas that are primarily limited by temperature. The yellow reaches are areas that are primarily limited by density of juvenile salmonids. And that's also, of course, a density physical habitat relationship at those reaches. And then the orange shaded reaches have a kind of a combination of both temperature and density limitations. And of course, the yellow density, as Melody talked about, the yellow density related or density limited reaches are also density limited because of the temperature constraints on the downstream of the left-hand side of this figure. Juvenile Chinook that are, can't survive, can't tolerate the temperatures in those reaches are migrating upstream into this yellow reach, which is the Oxbow Conservation Area. And so you end up with a very high density of fish in some of those reaches where doing actions to create further physical habitat complexity could benefit the population there. Conversely, doing those actions, the physical habitat actions in the downstream reaches or other reaches that are flagged by the red shading here, is not likely to create a population impact until we first address that temperature. Next slide, please. So overall, I think our broad conclusion here is that despite worsening global environmental conditions, stream temperature and salmonid freshwater productivity have been maintained, but have not increased as we had hoped, as Melody showed you in that kind of hypothetical figure. And we didn't have time to get into all of the, hundreds of figures from the 15-year report here, but one key take home for me, I think, is that we've documented that air temperatures in this area have risen since the inception of the IMW. But despite the rising air temperatures, stream temperatures at many of our monitoring sites have remained stable. So the stream and riparian recovery that's occurred to date has thus far sort of held a line, held stability in stream temperature despite the increasing air temperatures. And so that's also translated into thus far stable population abundance, although we haven't achieved or come close to achieving what Oregon would call a broad sense recovery of these populations. But at least we have not declined into a conservation hatchery or more drastic intervention type of approach. Next slide, please. And so again, some broader scale context here I think is important. ODFW had a research project for Chinook in the John Day Basin from the mid-1970s until the early 1980s. And here are just a couple of quotes from that report, the completion report that was done in 1985. So at that point in time, returns for spawner in the Middle Fork had been below replacement since the 1976 brood, suggesting that after a short period of adjustment, the Middle Fork stock is now also declining. And then the report further went on to say that if these trends continue, spring Chinook salmon in the North Fork, historically the most productive stream in the basin, will be reduced to a remnant run in less than 30 years. But here we are, about 40 years after that report was completed, and those populations have largely, there's been ups and downs for sure, but they've largely remained stable. And again, like I said, we haven't had to resort to any sort of conservation actions or hatchery type programs. So I think that's a positive outcome from the collective effort of riparian and stream restoration work in the Middle Fork IMW. But certainly, again, not anywhere near the broad sense recovery that we want to be. And I think to get there, we really need to consider the key takeaway from the Middle Fork IMW is that restoration actions must occur in a reach scale context. And that's what we're trying to convey and provide via those color-coded stream network maps. Next slide. And so within the context of each of those reaches, the habitat actions have to address the reach-specific limiting factor. And we also have to realize and acknowledge that it's likely to take multiple work iterations because, when we say that stream temperature is a limiting factor, we also recognize that we can't get there with just a simple approach of adding vegetation or adding wood. We may need to do extensive channel reset actions in order to better merge all the historic hydrology and floodplain connectivity processes that have been lost, and then get to a state where we can work on the stream temperature limitation. So it's likely to take multiple actions or multiple iterations to get there. And that's not, can't be viewed as a bad thing. It has to be viewed as a iterative process where we're working and learning through time. And it's just going to take multiple steps to rebuild the historic productivity of some of these stream reaches and populations. Next slide. And so really at 15 years into the IMW here, I think it's also important to assess our position in the adaptive management loop and continue to reevaluate whether answering the original question about salmonid productivity is the only direction for this IMW and this IMW group. And Kasey and Melody and others have talked about this a lot. And I think really we're at a point here now where we need to begin shifting to a proactive and structured restoration processes through really intentional engagement and demonstrating the co-benefits of restored floodplains with socioeconomic needs, both within the Middle Fork and also at the much broader regional scale, to transfer these lessons to a broader scale and begin to set the stage for restoration occurring at a broader scale as well. Next slide. So with that, I just think we want to broadly acknowledge everyone, the numerous people who've contributed to this work, and then I think we can jump over to the next slide and we're ready to field questions. So thank you everyone for taking time to listen, and looks like there are several questions in the chat already. So Meg, if you want to queue those up, we can start working through those.
Yeah, totally. Great presentation, guys. All right. First out of the gate, if I'm not missing anything, is Jen O'Neill. Was all snorkeling done during the summer, during the day?
Yeah, I can tackle that one. They did occur during the summer, during the day, and For the second part of your question, would seasonal differences be likely to affect Chinook par? I think what we find is that once par are somewhere for the summer, they don't really move until flows improve. So I don't know if it would be different in the fall, and then in the spring. In the spring is either when they're kind of out migrating as smolts or they're as fry. And so, yes, I think they would be seasonal differences, but I would also say as Chinook move through their life cycle, they're experiencing different conditions and they're just at a different part throughout seasons. We definitely do think it could vary from year to year. For example, when these results, and the year we did this project was 2021 during the heat dome year. And so we're not quite sure, but we thought that might have pushed more par into the tributaries versus the main stem in such a hot, maybe kind of drier year. We might see more in the main stem during a cooler, wetter summer. So.
All right. And Kasey Justice, I see your hands up out there. I'm going to tackle a couple of these early ones, and then we'll have you unmute. All fry migrate downstream, and a proportion of them move upstream is par? Question mark.
Yeah. Yes, but it's kind of hard, I guess, because we have to remember that they were collected as snapshots in time. So some were from the par life stage and some were from the fry. So it's not necessarily the same fish that move upstream and downstream. It could have been fish that started downstream and moved upstream. Yeah, we don't have like that continuous snapshot. It's just snapshots in time. So I hope that answered that question. But essentially, yes.
All right. Question from Ray. Any opportunity for riparian improvement to help cool waters in some areas?
Yeah, I did mean to touch on this. Yes, there is opportunity for riparian improvement, and that's kind of our main strategy for cooling water temperatures. But we're also trying to look into other avenues. We have a project proposed that we're going to look into kind of more on a microhabitat scale where we can see temperature changes, and we're hoping that we'll come up with some strategies like implementing certain habitat units and restoring hyperrheic flow, because riparian improvement can take so long. We're hoping for some kind of short-term restoration tactics that we can also employ to cool water temperatures sooner than growing trees. So,
okay,
all
right.
I can add. I can add on to
Kasey.
that for a minute too, Meg. I didn't include the restoration lessons learned and themes around recommendations, but riparian, like Melody said, riparian strategies and riparian management have been a big focus, and there's been a lot of study around planting and some planting failures in the Middle Fork, and there was a whole group that met in the end of 2023 to put together a riparian planting strategy, and that is included in our 2024 report, and to really focus in on that and help restoration practitioners really learn and help with this riparian planting. So there's a lot of talk and focus around riparian strategies.
Okay, great answers, guys. Jen O'Neill, I'm gonna do your follow-up after Kasey. So go ahead, Kasey.
Hi, thanks. Really awesome presentation. I have a question for Melody. You were going over the slide that showed kind of the annual trends in spawner abundance over time and how we haven't really seen the improvement that we were hoping for. And I was wondering how kind of average temperatures in the main stem have changed over that same time scale, and whether there may be a warming trend that's potentially masking benefits from restoration. Thanks.
Yeah, good question. There is a chapter in the report by Stephen Kelly on temperature, and kind of his major findings or takeaways are, even with warming air temperatures, it seems like many sites have some stability. I would say something that is possible, especially in adults, is that they're also seeing ocean effects that we can't control. And so, since we are starting to see some maybe slightly positive, slightly less negative results in the in the Middle Fork, or, from freshwater productivity, that's where those fish aren't experiencing those oceanic conditions. But I don't think in the main stem we've seen extensive rising temperatures in the Middle Fork. I think, again, with temperatures, we've generally seen stability, even with rising air temperatures.
Okay.
If Kasey or Ian have thoughts on that, please chime in too.
Thank you.
Yeah.
Kind of watching Ian and Kasey's mics. Okay, I think you guys are good. All right, Jen O'Neill had a follow-up question. She's interested in last statement about the socioeconomic and floodplain relationships. Why and how to measure them?
Yeah, that's a good question. Thanks for that. I don't think we have a clear-cut, approach yet on how to quantify those, but I think the goal, that's come from a lot of discussions with Chris Jordan and others. I think a goal is to make this exportable and a sociologically feasible technology, right? If we can only restore floodplains and do these habitat projects in areas that are set aside as conservation properties, then that, likely precludes our ability to restore or, uplift a lot of the populations throughout the interior Columbia. But if we can demonstrate, whether it's through a qualitative or potentially quantitative approach, that you can do these floodplain projects and still have some of the benefits to socioeconomic needs throughout those same reaches, then that potentially creates access to a much broader portion of the riverscape that's not, set aside just as a conservation property. So yeah, that's a good question. I think we're at the beginnings of that, of trying to get there, and don't have a clear, answer for you on that right now.
Okay, and I see, Kasey, you had trouble getting to the 2024 report. I'll drop a link in a minute, or if somebody out there has the link open, specifically to that report. Kasey, do you have it?
Yeah, Jen, Meg. If you follow, I'll put the link in there, but if you follow the QR code, that will take you to our ODFW hosted data clearinghouse website that has all of our 2024 reports. It's also, thank you, Lauren. It's also on OWEB's website. And I was just responding to Kasey Justice to say that we don't have it uploaded to the IMW website right now. I'm working on some big updates on the IMW website, so it will be up there soon. But for now, you can find it at OWEB. You can find it at that QR code.
Okay. All right, perfect. And then I'll send out a link too in the follow-up email too to help you guys out. And Julie Furman had just made a comment: restoring hyperic flow can be more effective than riparian shading too, just not a short-term band-aid. Right, Jen O'Neill, looks like there was a bump in Chinook red densities in 2014 through 2016. Any ideas on the bump? Clearly a lot of good data here.
I think Melanie answered that.
My mic was muted, but I also wanted to say that I totally agree on the hypereic flow comment. Yeah, definitely not just a short-term band-aid, but there was a study done about 10 years ago that found very little hypereic flow in the Middle Fork, but it hasn't been looked at since some extensive restoration has occurred. So we're hoping that possibly we actually are unclear what the literature suggests about hypereic flow being restored, and we're not sure if it's just a characteristic of the Middle Fork or if it's due to such degrading conditions and alterations from mining and that kind of a thing. So, but yes.
Awesome. Ben Staten had a question. In the juvenile production time series figures you showed, have you considered evaluating productivity in terms of juveniles per spawner rather than total juvenile production?
Yes, that actually tees up a really good thing that I forgot to cover during mine about the future of our genetics project. Our next, we kind of shifted that project to where we collect our juvenile genetics. We're going to start collecting them at the smolt trap so that we can start looking at juveniles per spawner, and then the success to the smolt life stage. And then we'll look at those metrics. I don't think for the data that we've already collected, I think we've looked at it, but we haven't quite done anything with it at this point. So that project is also still ongoing and changing to look at kind of survival by red location and spawner. We do have that included in that sort of watershed scale report. So we did look at juveniles per spawner, and did some residual analysis on that. So it's in that first chapter, the watershed scale. Yeah, I also want to say there are so many more results in the summary report, but we were only able to just scratch the surface today.
Totally.
Awesome. Excellent questions, everybody in the crowd.
Yeah.
Right now, we're just kind of, I don't see any more questions. If anybody wants to unmute and say anything. You guys have got them cornered here. You can ask them all the questions you want. Go ahead, Julie.
Yeah, so this is really great, guys. Thank you for sharing all of this. So in the question about the lack of detection of a change, it seems like from what you presented that there's maybe at least 3 different possibilities there. One being that the restoration that has taken place hasn't had enough time to mature, or possibly that there hasn't been enough time for the fish to respond to the change in the restoration. You mentioned too that maybe something needed to be shifted in your monitoring to detect changes that might actually be there that you haven't seen. And I can't remember the third thing you mentioned that, but I was just curious if you have a gut feeling about which of those is likely to be true, or if you talked about some you're monitoring going forward, targeting specific questions, if those are some of them, if there are others. I'm just interested in learning more.
Yeah, those are really good points. I think a couple of them that maybe we didn't touch on was that maybe not enough of the watershed has been restored. So we're not seeing those population level changes. And I would also say a big one is that we don't think we've addressed the limiting factor, which is temperature. So a couple of those reasons we think, but and I think that's, we're still going to keep doing that watershed scale monitoring, but we need to focus our restoration to smaller scales. So those like reach scale levels, and try to see improvements to specific restoration actions versus this is how much the watershed has been restored and this is the results we're expecting to see. Like we need to like kind of adjust our scale, and that's what we're trying to do.
Cool, thanks.
Yeah.
Yeah, Julie, I also, as Melody touched on too, and something we've talked about in the IMW is, there was, restoration is very much ongoing there. And as Ian also mentioned, there's changes as we're learning across all restoration projects across the Columbia Basin and also within the IMW about these iterative processes. So this, we had our annual meeting with John Day Basin Partnership last month, and we went out to see the Oxbow Conservation Area, which has gone through 5 phases of restoration and is on to phase 6. And they were pointing out places where they had done some restoration actions really early on, and those now they're discovering are inhibiting, say, maybe their plant growth and not allowing floodplain and water to, get out to the plants. And so they're going to start changing some of that. So I think part of it is also that learning process. This was a new and kind of a big idea, and just kind of keep stepping through it. So.
Really. All right. Jen, go ahead.
I have a question. Who's the hardy soul in the photo? I love the winter photo.
It's Melody.
Awesome.
I was admiring that as well.
That is what you get when you do fry sampling. So.
Excellent. Yeah, looking warm and toasty there, Melody.
We actually tried to snorkel sometimes too for fry. It was freezing and it didn't really work. So now we just dip net for them.
Excellent. All right. Well, we've got about 4 minutes left. I think Ian and Kasey and Melody are willing to take emails too if you guys come up with some brilliant questions post meeting. So they're available for you guys, and I'll make sure to kind of give out contact info and whatnot in the follow-up email. Otherwise, thanks so much to Kasey Bliesner, Ian Tattam, and Melody Feden. We really appreciate you guys presenting this today. I think a lot of people learned a lot, and it was really helpful to kind of deep dive a little bit with you guys. I will wrap up with some reminders about how to access fish monitoring workgroup information. Our website has a refreshed look, so go check it out. Jen will drop a link in the project page in the chat again, just in case you missed it earlier in the meeting. If you'd like to be added to the distribution list, or if you have some interesting work you'd like to present or have a presentation recommendation, let me know. We have a resource. We are a resource for you guys, so let us know if you have suggestions. We're contemplating changing the reoccurring meeting time and third Thursday sequence in 2025. So look out for a poll link in the follow-up survey just to see if maybe a different time setup would work for you guys. Otherwise, the recording of this meeting will be available next week. The group will convene again in January. To close out, I would like to give a big thank you to Kasey, Ian, and Melody for presenting, and a thank you to our core team, task leads, and you guys, our work group. So enjoy the rest of your week.